Hi, and welcome to week 4 of Applied Immunology. This is lecture 4A, which begins the week 4 module where we will transition into discussing components of the adaptive immune system. We will start off with our talk today on the mechanisms that govern the generation of lymphocyte antigen receptors, which again are B cell receptors and T cell receptors. We will cover the structure of these receptors and also discuss the unique mechanisms of genetic rearrangement and modification that occur which yield huge repertoires of BCRs and TCRs with unique specificities. Since this lecture establishes the structure and generation of receptors for both T and B lymphocytes, it's kind of a bonus lecture for this week, so we'll have a bit more total lecture time during week 4 of this course. Also, please note that the remaining lectures for week 4 will focus primarily on T cell biology, while week 5 of this course will focus on B cell biology. We've seen this schematic a few times by now, which summarizes the activation of the two main subsets of lymphocytes, B cells and T cells. We know that B cells express a B cell receptor, or BCR, as their antigen receptor, and that this can bind to soluble antigens. We also know that BCR activation can lead to the proliferation and activation of B cells. An important naming convention for these proteins is that the membrane-bound form is called the BCR, while the secreted form is called an antibody, and the protein structure of both forms is called an immunoglobulin, and this is usually abbreviated as Ig. We also know that T cells express a T cell receptor, or TCR, as their antigen receptor, and that this binds to antigens that are presented as a complex of peptide plus MHC on the surface of antigen-presenting cells, or APCs. Like B cells and the BCR, TCR activation usually leads to the proliferation and activation of T cells. As a side note, in these schematics I usually just show a single BCR or TCR per cell type for the sake of simplicity, but in reality, each lymphocyte expresses many copies of the same antigen receptor at its surface. From our introductory lectures, we've established that a key hallmark of the adaptive immune response is a high degree of specificity for pathogens, and that this is achieved through the lymphocyte antigen receptors expressed on either of these cell types. Before we get into learning how immunoglobulins and TCRs are diversified to enable this level of specificity at the genomic level, let's first learn about what these proteins look like on a structural level. Let's start with immunoglobulin. These are Y-shaped molecules that consist of a constant region in blue here on the left, which remains relatively fixed between immunoglobulins within a shared class called an isotype, and we will discuss isotypes later on in week 5. The constant region makes up the central stem of the immunoglobulin, which is at the protein's carboxy terminus, as well as the base of the two branching arm domains that are fixed to the stem. The constant region is involved in the recruitment and activation of cells such as phagocytes, which perform effector functions such as phagocytosis, and again, we will discuss that a bit further in week 5 of this course. In addition to the constant region, immunoglobulins also have a variable region that is depicted by the red domains in this left diagram, which you can see are located in the protein's amino termini that are located at the tips of the branching arms of this Y-shaped protein. As we will find out shortly, the variable region is the part of the protein that recognizes cognate antigen, shown here by this little gray circle abbreviated as Ag. And therefore, this is the portion of the immunoglobulin that must exhibit high genetic diversity. The domains of the protein are named for each of these regions, with variable region domains being denoted by V, while constant region subunits are denoted by C. Another structural component of the BCR is that it is comprised of two types of protein chains, heavy chains and light chains. The heavy chains are the hinged portions depicted in green here on the right, and contribute domains towards both constant and variable regions of the protein. The heavy chains also link together the two halves of the BCR through disulfide bonds here in the center of the complex. The light chains are depicted in yellow in this diagram, and like the heavy chains, they contribute both constant and variable regions, although the light chains do not make up any of this central stem portion of the BCR. The light chains are connected at the base of the arms of the BCR through disulfide bonds with the heavy chain. Light chains come in two different types called kappa and lambda, and a single immunoglobulin molecule will only have either both kappa light chains or both lambda light chains, so they're never mixed between the two halves of the same immunoglobulin molecule. The heavy and light chains are also involved in the nomenclature of immunoglobulin subunits. So if you look back at the blue and red diagram on the left, you see that subunits are named using a V or C, which stand for variable or constant regions, followed by a subscript H or L, which stands for heavy or light chains. In the case of constant region heavy chain subunits, a number from 1 to 3 is also added. So we can refer to specific domains of the immunoglobulin using this nomenclature. For example, the names of the two domains involved in antigen binding would be the VH and VL domains, standing for variable heavy chain and variable light chain, respectively. One last note is that the immunoglobulin molecule can be split up into different fragments, including the FAB fragment, which consists of just one arm of the Y-shaped protein that has been disconnected from the stem of the constant region. And this is shown by the red dotted line in the immunoglobulin molecule on the right. The FAB fragment contains the variable region of the immunoglobulin and is actually quite structurally similar to the T-cell receptor. Now, compared to immunoglobulin, the TCR is a bit simpler in terms of structure. Both consist of two different protein chains, which in immunoglobulin are light and heavy chains, but in the TCR these are alpha and beta chains, and these are represented by the yellow and green colors in this diagram on the right. While immunoglobulin is made up of two heavy chains bound to two light chains, which together form two antigen binding sites, the conventional TCR is just one alpha chain paired with one beta chain through a single disulfide bond, and this only forms one binding site for peptide MHC. Similarly to immunoglobulin, the TCR also has a variable region at its tip that is made up by a portion of both alpha and beta chains, and this is also the site of the TCR that must exhibit the greatest amount of diversity in order to bind to an enormous repertoire of potential peptide MHC complexes. The variable region is connected to a constant region, which is made up of both alpha and beta chain segments. The constant region of the TCR connects to domains that insert through the plasma membrane and stick out into the cytoplasm. And these cytoplasmic tails of the TCR are important for signal transduction events which underlie T-cell activation, which we will discuss later on this week. Like immunoglobulin, the TCR subunits are named according to which chain they belong to and whether they are part of the variable region or the constant region. Again, this is abbreviated with a V or C for variable or constant, respectively, and then alpha or beta referring to the alpha or beta chains of the molecule, pretty obviously. So the four extracellular domains of the TCR are V alpha, V beta, C alpha, and C beta. It's worth noting again that when we compare the structures of immunoglobulins and TCRs next to each other, variable and constant regions of the TCR look very similar to the FAB fragment of immunoglobulin. So the TCR essentially looks like a membrane-bound form of the FAB fragment of immunoglobulin, although there are some differences in the C-alpha domain of the TCR and how the domains of the TCR interact with one another. However, let's just try to keep some of these overall similarities in mind once we start to learn about where variability is localized and how genetic diversity is achieved for both immunoglobulins and TCRs. Before we get into genetics, let's develop our understanding of precisely where variability is found in the structure of lymphocyte antigen receptors. The variability encoded in the antigen binding portion of lymphocyte antigen receptors can actually be mapped to specific segments of the protein called hypervariable regions, or HVs. In this example, let's consider an immunoglobulin molecule where the variable region is made up of both heavy and light chain proteins. These diagrams show something called a variability plot, which compares the amino acid sequences of several different immunoglobulins across both the heavy and light chain variable regions, again denoted by the letter V. The position of each residue is plotted on the x-axis, while the y-axis represents a ratio of the number of distinct amino acids observed at a given position across all sequences to the frequency of the most common amino acid. And this might sound a little bit complicated, but don't worry about it. It simply serves as a measure of residue variability. It's obvious from both of these plots that there are three distinct spikes highlighted in red, which represent the hypervariable regions. There are 3 each in the heavy chain and 3 in the light chain, and these are creatively named HV1, HV2, and HV3. These regions are separated by framework regions, denoted as FR. Importantly, these hypervariable regions are responsible for forming the surfaces at the tip of the variable region that actually bind to foreign antigen. This last point is easier to illustrate when we map the amino acid sequences of these domains onto actual protein structures. From the central diagram, it's obvious that the three red hypervariable regions of the immunoglobulin light chain variable region are separated sequentially by framework regions, or FRs. Let's first look at the flattened ribbon diagram at the top panel on the right here, which shows that this protein domain is arranged as a series of 9 peptide strands that alternate running in opposite directions. And this is a biochemical structure called antiparallel beta sheets, or antiparallel beta strands. Notably, although from the sequencing diagram in the center it looks like the hypervariable regions are separated from one another by framework regions, when we map these onto the flattened ribbon diagram, we find that the HVs actually occur in the loops which join the beta strands. And in this configuration, the HVs actually end up on the same top portion of the molecule, even though they are sequentially separated by framework regions. Moving to this middle panel on the right, this represents the three-dimensional folded structure of the variable light chain domain. Here you can see that the beta sheets roll together to form something called a beta barrel. This is a roughly cylindrical shape, where all of the residues that form the hypervariable regions in red are clustered together at the tip of the protein, where they form a binding surface for cognate antigen. The bottom panel on the right simply shows where the hypervariable regions are located in the grand scheme of an entire immunoglobulin molecule, and this is indicated by the red tips of the variable region, which forms the antigen binding site. Keep in mind that this schematic is only showing the domain structure of the light chain variable region, which is paired with a heavy chain variable region to make up one side of the Y-shaped immunoglobulin protein. Therefore, the antigen binding site for one half of the immunoglobulin is made up of 3 hypervariable regions from the heavy chain plus 3 hypervariable regions from the light chain. Since these form a surface that is complementary to foreign antigen, these 6 hypervariable segments of the protein are commonly referred to as the complementarity determining regions, or CDRs. Together, these loops of CDRs from both the heavy and light chains are also called the immunoglobulin fold. Keep in mind that this is just on one of the Y-shaped arms of the protein, so each immunoglobulin molecule has 2 separate antigen binding sites, one on each tip of the Y. Now, this example is centered on sequences and structures of an immunoglobulin molecule, but keep in mind that the variable region of the TCR exhibits similar types of hypervariable sequences with some key differences. The TCR contains hypervariable regions that are named according to whether they belong to the alpha or beta chain, and like with immunoglobulin, these are also referred to as complementarity determining regions, or CDRs. Notably, while the antigen binding site of immunoglobulin variable domains is determined by 3 CDRs, T-cell receptors actually have 4 CDRs in their sequence. However, TCR variable domains exhibit different 3-dimensional structures compared to variable domains of immunoglobulins. So in TCRs, the CDRs are not actually clustered together at the tip of the molecule the same way that they are in immunoglobulin variable domains. Instead, the third subunit on the alpha chain, called CDR3 alpha, is the primary hypervariable region involved in binding to peptide MHC. But other than that, the TCR and immunoglobulin are similar in that their variability is concentrated within fairly small ranges of amino acids in their overall protein sequences. Now that we understand what the lymphocyte antigen receptors look like and which parts of those proteins are involved in antigen binding, let's next learn about the mechanisms that are used to generate diversity at these binding sites from the genetic level. A common theme that we've hammered home so far in this course is that the adaptive immune system must have an enormous amount of potential receptor specificities in order to be prepared to respond to any possible foreign peptide sequence. The average adult is estimated to have approximately 2 trillion T and B cells, and hypothetically, each of these cells express their own unique lymphocyte antigen receptor. Given that the human genome has a relatively limited size, where it only encodes roughly 20 to 25,000 genes total, how are these numbers of unique antigen receptor genes even possible? So there are 3 mechanisms shared between both B and T cells that we will learn about today, which allow this to happen. And we will discuss each of these in more detail shortly, but let's first look at a basic summary of these processes. The first is a form of combinatorial diversity that is achieved through the splicing together of assorted variable, or V, diversity, D, or joining, J, gene segments, which make up the CDRs of antigen receptors. Each of these segments come in many different forms, and any V region can pair with any D and any J region through a process of genetic rearrangements called VDJ recombination. VDJ recombination is responsible for forming many different combinations of individual V, D, and J gene segments, which encode the CDRs of the antigen binding site, and this is why we refer to this as combinatorial diversity. This is accomplished through interactions between recombination signal sequences, or RSSs, that interact with the DNA recombinases RAG1 and RAG2, which are followed by the activities of a complex of proteins that are involved in DNA double-stranded break repair, which include Ku, DNA PK, and something called Artemis. The second is junctional diversity, which is a component of VDJ recombination. Following rearrangement of V, D, and J segments, an enzyme called terminal deoxynucleotidal transferase, which is thankfully abbreviated as TDT, introduces random additions of nucleotides at the junctions between each segment. An exonuclease can also remove random nucleotides at these junctions. DNA ligase 4 and a DNA repair protein called XRCC4 are then responsible for joining the modified ends of these DNA junctions to complete gene rearrangement. These random nucleotide alterations contribute massive amounts of sequence diversity, specifically at the junctions between gene segments that encode the antigen binding site for these receptors. The third is yet another form of combinatorial diversity that is achieved through the pairing of different antigen receptor subunit chains. So if you remember what we just learned about antigen receptor structure, following completion of the genetic rearrangement processes outlined in points number 1 and number 2 here, a B cell could pair any rearranged heavy chain with any rearranged light chain to make its own unique immunoglobulin molecule, while a T cell could pair any rearranged alpha chain with any rearranged beta chain to make its own unique TCR. Note that unlike VDJ recombination, this form of combinatorial diversity is not accomplished through molecular mechanisms at the DNA level, but instead this occurs at the protein level by linking different protein chains together to form a complete receptor. Because this second form of combinatorial diversity is pretty straightforward, for the rest of today's lecture we will focus our discussion on learning more about the molecular genetic mechanisms of diversity that are outlined in points number 1 and number 2 above here. As a last side note, there is a fourth mechanism of antigen receptor diversity called somatic hypermutation. However, this is only observed in B cells once they've become activated by cognate antigen and enter something called a germinal center reaction. So we're going to save our discussion of this process for the B cell module during week 5 of this course. Also note that there are additional processes that confer diversity in the constant region of immunoglobulin molecules, which we will also cover in week 5. Today, we will center our focus on the molecular mechanisms that are shared between both B cells and T cells in the generation of diverse antigen receptor repertoires, which again are the first 2 points outlined in this summary slide: combinatorial diversity of V, D, and J regions, paired with junctional diversity that is generated during gene rearrangement. Let's learn more about each of these, starting with the combinatorial diversity conferred by VJ or VDJ recombination. We will begin by looking at this process in the generation of immunoglobulin molecules that are expressed by B cells, which again consist of light chains paired with heavy chains, though keep in mind that identical processes occur in homologous alpha and beta chains in the TCR. The light chain on the left here, which comes in either kappa or lambda isoforms, consists of variable segments, or V, shown in red, joining segments, or J, shown in yellow, and constant segments, or C, shown in blue. So note that there are no diversity, or D, segments found in the light chain. Multiple forms of each gene segment are expressed in the germline DNA of every lymphocyte, and during development, each individual B cell will randomly pair a single V with a single J in order to generate the CDRs that are used to make its own distinct light chain variable region. This figure on the right illustrates where CDRs are found in these 2 gene segments that make up the variable region of the light chain. So CDR1 and CDR2 are derived from the V segment, while CDR3 is derived from the junction between V and J segments. Note that the constant regions of the light and heavy chains do not contribute to foreign antigen binding, and that C region splicing is controlled during transcription. Again, we will discuss how constant region variability occurs in B cells during a process called isotype switching during week 5. First, somatic recombination occurs during B cell development in the bone marrow. This recombination joins a single V region together with a single J region to encode the single VJ variable region sequence for that given B cell. And when the B cell needs to express its immunoglobulin, this lymphocyte gene is then transcribed into RNA. And it is at this transcriptional level of messenger RNA where another splicing event occurs that brings a single C region to meet with J. And this forms a transcript that encodes a VJC sequence for that B cell. This transcript is then translated to make the corresponding light chain polypeptide used to make immunoglobulin. Now, the heavy chain used to make immunoglobulin follows a pretty similar generation process compared to the light chain, with the addition of a diversity or D segment shown in green. The D segment is just an additional gene segment that is incorporated with V and J regions to make up the variable region that serves as the molecule's antigen binding site. Like with V, J, and C, the D segment also exists in multiple forms in the germline DNA that can be mixed and matched. So during development, each individual B cell will randomly pair a single V, D, and J region together in order to make up the CDRs that are located in its own distinct heavy chain variable region. Heavy chain recombination differs from light chain recombination in that the first somatic recombination event that occurs at the DNA level is the joining of a D and J segment together. The next step also occurs at the DNA level, where a V segment is joined with the existing DJ segment to encode the single VDJ variable region sequence for that given B cell. The C region is then added at the level of transcription when mRNA is processed to splice a single C segment together with the genomic VDJ segments. This generates a final transcript that encodes a VDJC sequence for that B cell's heavy chain, and this is then translated to make the corresponding heavy chain polypeptide used to pair with light chain proteins and yield a final immunoglobulin molecule. Note that the C region used to make the heavy chain can be changed permanently during a process called antibody isotype switching, which we've referred to a couple of times by now, but again, we will discuss this more in our week 5 lectures on B cell activation and effector functions. The combinatorial diversity that we've talked about is due to the fact that lymphocytes express high numbers of different types of each gene segment. These numbers of functional gene segments are summarized in the table on the left, while the genetic organization of these loci in the germline human genome are represented by the schematic on the right. Again, variable gene segments are in red, diversity segments in green, joining segments in yellow, and constant in blue. The large number of gene segments encoded by each of these loci means that they end up representing large portions of genomic DNA. For example, the heavy chain locus is 2 megabases or 2 million base pairs long. From these figures, it's obvious that gene segment numbers are highest in the variable segment for both forms of light chains, kappa and lambda, as well as in the heavy chain. Again, the light chain has no diversity segments, while the heavy chain can pick from any of 23 possible segments. Joining segments have the lowest diversity out of the segments that make up the variable region, while the number of constant segments is also relatively low across both heavy and light chains. Note that these numbers are derived from sequencing data for one individual, so the exact numbers of functional gene segments may vary slightly between individuals due to genetic polymorphisms. But at any rate, the number of possible combinations of each individual gene segment being paired with another starts to scale up pretty quickly, and this is why this source of combinatorial diversity represents an important contribution towards the generation of a diverse antigen receptor repertoire. Now that we understand the combinatorial diversity associated with VDJ recombination, let's get into some of the specific molecular mechanisms involved in these forms of genetic recombination. This is actually a really interesting process from a genetic standpoint, and again, I want to stress that lymphocytes are the only cell type in the body that can undergo these mechanisms of gene rearrangement. We don't really have the time in this course to fully dive into it, but I still want to provide some level of mechanistic detail regarding VDJ recombination. This example here is looking at V and J recombination in the light chain of immunoglobulin, but note that these processes are the same for VDJ recombination in the heavy chain of immunoglobulin, as well as in homologous structures in the T cell receptor. First, All of the V, D, and J gene segments are flanked by conserved non-coding DNA sequences called recombination signal sequences, or RSSs. These RSSs contain conserved heptamer and nonamer sequences at either end, and these are separated by a non-conserved region comprised of either 12 or 23 base pairs. I know that these numbers may seem very specific and kind of random, but they actually roughly correspond with either 1 or 2 turns of the DNA double helix, respectively. The RSSs are only found immediately adjacent to V, D, and J gene segments, where they serve kind of as flags for sites of recombination. It's also worth noting that an RSS with a 12 base pair spacer can only associate with another RSS with a 23 base pair spacer. And the distribution of these different length spacers amongst gene segments is what's actually responsible for ensuring that a V region can only recombine with a D or J region. So the 12-23 rule prevents a cell from accidentally joining 2 V regions together or 2 J regions together, etc. In the diagram at the top, the RSSs are represented by the purple triangle next to the red V segment and the orange triangle next to the yellow J segment. And the diagram at the bottom just shows the same step at the level of nucleotides. The reason why recombination is restricted to pairs of 12 and 23 base pair spacers is through the activity of this RAG1 and 2 recombinase enzyme, which will only bring 12 base pair and 23 base pair RSSs together for its enzymatic activity. RAG, which stands for recombination activating gene, is an endonuclease that's only expressed in lymphocytes, and it's responsible for splicing out intervening DNA to join V and J segments as step number 2 in this process. Just as a kind of interesting evolutionary side note, the RAG enzymes are thought to be derived from an ancient virus that encoded a DNA transposase. It's proposed that this virus infected an evolutionary ancestor of vertebrates, which then co-opted this recombination activity and therefore gained the ability to start generating lymphocyte antigen receptors and therefore an adaptive immune system. This likely provided such a survival advantage that the viral enzyme became fixed in the vertebrate ancestor's genome, and this is thought to be why all vertebrate lineages have an adaptive immune system. So anyways, in this diagram on the top, you can see that RAG1 and 2 in purple bind to one RSS, which is the light purple triangle adjacent to a red V segment. And upon binding, RAG1 and 2 then recruits a second corresponding RSS, which is the orange triangle adjacent to the yellow J segment. And this brings the V and J segments together at the same site in order to target both with the endonuclease activity of the RAG complex. RAG cuts the DNA to remove the RSSs from the target gene segments, and the cut ends then form a DNA hairpin at the tip of a blunt double-stranded DNA break. Following RAG1 and 2 cleavage of genomic DNA, 2 joints are formed. There's the coding joint, which is associated with the genome and shown in the left column, and then something called the signal joint, which forms an extra chromosomal loop of DNA that is lost following cell division, and this is represented in the right column. We will focus primarily on the coding joint for the remainder of this discussion, since this is what will actually eventually encode the lymphocyte antigen receptor. The coding joint consists of DNA ends that were cut by the RAG complex, each of which form a hairpin structure. At this stage, the recombination site can then recruit a series of proteins associated with double-stranded break repair, and this is a form of DNA repair that's used ubiquitously in somatic cells. So keep in mind that unlike RAG1 and RAG2, these DNA repair proteins are not only expressed by lymphocytes. The third step in this process involves the recruitment of a repair enzyme called Ku, which is made up of Ku70 and Ku80 subunits. These form a heterodimer with a ring or donut-like structure, shown by the green loops in this diagram, and these rings then sort of cap the DNA hairpins. Ku is responsible for holding the hairpin DNA ends together, and it's also responsible for recruiting the next enzyme in step number 4 of this process, which is called DNA-dependent protein kinase, or DNA-PK, and this is represented by this light purple blob in the diagram. DNA-PK then recruits a nuclease called Artemis, which is the dark purple enzyme in this diagram. Since DNA-PK is a kinase, it functions by phosphorylating Artemis, which activates its nuclease activity. Artemis then nicks open the DNA hairpins at the blunt ends of the DNA that needs to be recombined. This nick can occur randomly at any of several nucleotides involved in the hairpin, which also contributes to sequence variability. This nicking then yields single-stranded DNA ends, as shown by this diagram on the bottom. Since these are endogenous nucleotides that were originally found at the intersection between the RSS and the target gene segment, these often form palindromic sequences once the hairpin is nicked open. And these are therefore called P nucleotides, and they are highlighted in the light blue shading in this lower panel figure for step number 4. Single-stranded DNA breaks can then be targeted by an enzyme called terminal deoxynucleotidyl transferase, or TDT. This is step number 5 of the gene recombination process. Like the RAG1-2 complex, TDT is an enzyme that is only expressed in lymphocytes, so it's not a part of the DNA double-stranded break repair machinery that was involved in the previous steps. It makes sense that the expression of TDT should be restricted only for the process of antigen receptor diversification, and this is because its activity is to add random nucleotides to the ends of single-stranded DNA breaks. This is a major source of junctional diversity, which is the second form of antigen receptor diversity that we introduced today. Obviously, the expression of this enzyme would need to be restricted only to lymphocytes during VDJ recombination, since introducing random nucleotides would be a very dangerous source of mutations in most other cell types. The nucleotides added to single-stranded DNA in this step are non-templated nucleotides that are tacked onto the end of the endogenous P nucleotides. And so these added nucleotides are abbreviated as N nucleotides, and they are highlighted in blue in this lower panel figure for step number 5. TDT can add up to 20 additional N nucleotides to existing P nucleotides on single-stranded DNA. And again, this is what we refer to as junctional diversity. Following N nucleotide addition, complementary nucleotides on either strand pair together, and any extra unpaired nucleotides are trimmed by an exonuclease. Importantly, exonuclease activity can also remove several nucleotides if pairing does not occur, which means that both TDT-mediated nucleotide addition as well as exonuclease-mediated nucleotide subtraction are two sources of junctional diversity during VDJ recombination. Following exonuclease activity, single-stranded gaps between the gene segment and paired regions of the junction are filled in using DNA synthesis and ligation enzymes. And these function by essentially repairing the coding joint. Enzymes involved in this process include DNA ligase 4 and the DNA repair enzyme XRCC4, which work with DNA polymerases mu and lambda to repair the single-stranded portions of this junction. The end product of this is what we refer to as an imprecise coding joint, denoted by step number 7, in which gene segments have been brought together, in this example the red V segment with a yellow J segment. This is done in an imprecise manner because junctional diversity has been introduced between each segment. Keep in mind that this process is repeated for every rearrangement that happens at the DNA level in lymphocytes. So this includes V to J joining in immunoglobulin light chain molecules, as well as D to J and V to DJ joining in heavy chain molecules. Also remember that similar processes occur in T-cell alpha and beta chains. The addition and deletion of nucleotides which join each target gene segment are the source of junctional diversity for lymphocyte antigen receptors. The total number of nucleotides introduced or deleted through these processes is completely random, and notably, some of these will result in a frame shift mutation that renders the resulting protein non-functional, and we refer to these as non-productive rearrangements. Almost 2 out of every 3 rearrangements is non-functional, although the remaining 30% of rearrangements which successfully form functional receptor proteins are the source of the enormous amount of diversity that we observe in lymphocyte antigen receptors. The example that we've followed so far in this lecture centers on the molecular mechanisms of receptor diversity in immunoglobulin molecules, but keep in mind that very similar processes occur in T-cell receptors. The molecular mechanisms of VDJ recombination are conserved between both immunoglobulins and TCRs, so all of the enzymes that we just talked about do the same exact things when making T-cell receptors. The genomic loci of the TCR are arranged similarly, so the alpha chain at the top here is homologous to the light chain of immunoglobulin, and that the variable region of the alpha chain is only made up of recombined V in red and J in yellow gene segments, which then connect to a constant segment in blue that does not contribute to antigen binding. The beta chain of the TCR shown at the bottom of this diagram is homologous to the heavy chain of immunoglobulin, and that it also contains D gene segments shown in green, and these make up the variable region of the T-cell receptor. An important difference worth noting between immunoglobulins and TCRs has to do with their constant regions. As we will learn in upcoming Week 5 lectures, the function of antibodies is highly dependent on the structure of their heavy chain constant region, as different variable regions of an immunoglobulin can be paired with a handful of different heavy chain constant regions that correspond to different antibody isotypes, each of which have varying effects on downstream immune responses. T cells, on the other hand, do not need the additional layer of diversity that's offered by isotypes, so they only relay one type of intracellular signal transduction cascade in response to peptide MHC binding by the variable region. So the locus encoding constant regions of the TCR is much simpler than that of immunoglobulin because there's only one constant segment used in the alpha chain and two possible constant segments used in the beta chain, which are extremely similar to one another. This is in comparison to the constant segments of the immunoglobulin locus, where there are 4 to 5 different C segments that can be used for lambda light chain proteins and up to 9 that can be used for heavy chain proteins. The number of gene segments directly corresponds to differences in functionality between the immunoglobulin molecule and the TCR. And we will learn more about this over the course of the next few lectures on adaptive immunity. This has been a really dense lecture, but I don't want you to get too bogged down in the molecular details of lymphocyte antigen receptor structure and generation. So let's refocus the take-home message of this lecture with this final chart, which summarizes the contributions of each of these mechanisms to antigen receptor diversity. We first learned about how high numbers of different V, D, and J gene segments can be paired together, forming all sorts of possible combinations, which represent the first mechanism of genetic combinatorial diversity, which we had listed earlier on in this talk. We've also learned about the process of P and N nucleotide manipulation when joining these segments in imprecise coding joints. And this adds an important second mechanism of genetic junctional diversity. So, considering these two molecular genetic processes, this table presents the total estimates summarizing how many different forms of immunoglobulin molecules and TCRs can be formed in a single individual. And this is really astronomical considering, again, that the human genome is restricted to about 25,000 genes. Again, these are the only cells in the body that are capable of undergoing genetic rearrangements like this. And you can imagine why tight control of these processes is extremely important in order to avoid introducing mutations in cells that could result in pathologies such as cancer. To summarize, today we've learned about the molecular mechanisms that allow for the generation of massively diverse repertoires of lymphocyte antigen receptors that are expressed by adaptive immune cells. These include immunoglobulins in B cells and T cell receptors in T cells. We've learned about the structure of both of these molecules at a higher level of detail and how they contribute hypervariable CDRs that make up the antigen binding site of these receptors. We've also learned about the unique molecular genetics of VDJ recombination in lymphocytes that allows for genetic rearrangements that generate huge repertoires of receptors. And we refer to these as providing both combinatorial and junctional diversity. Lastly, we've established that further combinatorial diversity is achieved through alternative pairing of different component protein chains in either immunoglobulins or TCRs. Again, remember that B cells also exhibit additional mechanisms of diversity which involve variation in the heavy chain constant region of the immunoglobulin protein, as well as something called somatic hypermutation in germinal centers, which we will discuss more in Week 5 during the B cell module for this course. That's it for this lecture, but please remember to watch the remaining 3 lectures for Week 4, which will cover remaining aspects of T cell biology and their functional contributions to adaptive immune responses.